We've probably spent quite a few classes discussing some important processes occurring in our nuclei, the control center of the cell. Each of these processes has come with its own list of important proteins, and we get it, there's been a lot to memorize. You've learned, for example, that during replication, helicase separates DNA strands, single-stranded binding proteins hold that separation in place, and DNA polymerase synthesizes new DNA. During transcription, you've learned of the RNA polymerase and a number of transcription factors that separate the DNA strands and add RNA nucleotides to the template, synthesizing an mRNA transcript. Some of you have even discussed chromatin remodeling by epigenetic factors, including acetylases and methylases that regulate gene expression. Nuclear processes are complex, and the proteins are many. But let's pause here. No biology class is complete without some discussion of these vital processes happening inside our cell's nuclei. These discussions are followed with equally important discussions on mRNA translation into proteins, a process that notably occurs outside the nucleus, in the cell's cytoplasm. Given this central dogma of biology, a glaring question remains. How do these proteins, synthesized in the cytoplasm, but necessary for processes occurring inside the nucleus, enter the nucleus? The question lends itself to another, broader question. How is transport, in general, mediated across the nuclear envelope? Our answer comes in the form of nuclear pore complexes, or NPCs. Proteins are imported into the nucleus via these NPCs, and they're also the main regulators of import and export between the cell's nuclei and cytoplasm. You've probably learned that the nuclear envelope encloses the nucleus with a bilayer lipid membrane, ensuring the spatial separation of the nucleus and the cytoplasm. Protein import into the nucleus is then anything but random, and the regulation of this import is emerging as a complex mechanism of regulating nuclear processes.
So, what is a nuclear pore complex exactly? Where is it, and how does it control what's going in and out of the nucleus? The nuclear pore complexes are these complex protein structures with around 1,000 individual protein subunits that are embedded in the nuclear membrane. Think of them as gates into the nucleus. Now, we can imagine that since the nucleus contains our DNA, typically something we don't want to mess with, we only want whatever's coming in to be what's supposed to be coming in. That is to say, we want these pore complexes to be pretty selective. Remember how I said nuclear pore complexes are kind of like gates to get into the nucleus? Well, as with everything in biology, it's a little more complicated than that. So, usually when we think of a gate, we think of something opening and closing. However, NPCs don't open and close to regulate transport. They're always open. Yeah, I know what you're thinking. How is this a gate then, and how are nuclear pore complexes being selective with what they let in and out? Well, they're special gates. Rather than being a traditional open and close gates, these gates are always open, but you have to show some identification to get through them. So, let's talk about how, molecularly, NPCs are allowing transport in and out. Alrighty, so do you remember how R groups interact, and that's super important for protein specificity? Well, we have a complex with around 1,000 individual protein subunits in each NPC, so this is an R group galore. Specifically, we have a unique phenylalanine and glycine repeat in the center of the pore complex. It's a lot of repeats, and it's pretty dense, so it makes almost a mesh-like layer. Think of this mesh as a very, very selective filter with only some things being able to get through. This leads us to the question of what these things, these molecules, actually need to show to get through these very special gates. So, this passage requires karyoparents, proteins that team up with nuclear-destined molecules in the cytoplasm and mediate their entry through the nuclear pore complex and into the nucleus. Together, the molecule and the karyoparent form a complex that's capable of interacting with and disrupting the NPC's mesh network so that it can go through it. It's complicated, but basically, molecules plus the nuclear transport factors get through the mesh. Molecules alone can't get through the mesh. What's so special about the newly formed protein complex that allows it to go through the mesh? I think before we talk about that question, we should slow down a little bit and talk about how we make the complex in the first place. Basically, how does the karyoparent find the nucleus-destined molecule in the cytoplasm? Well, let's recall back to foundations when we learned about protein zip codes. They direct proteins to different parts of the cell, like the mitochondria, the ER, and for our purposes, the nucleus. Since we're going to the nucleus, the specific name for this zip code is called a nuclear localization signal, or an NLS. Often, the NLS is hydrophobic, as it seems to be pretty important for karyopherins to recognize these cargo proteins. Once the two components come together to form an overall complex, they're now ready to go into the nucleus. Going back to the question of what about this complex is allowing it to go through the inner mesh found in NPCs? Currently, researchers have proposed a really cool model on how the complex disrupts the mesh. The two parts of the complex, the karyopherins and its cargo, individually can't go through the mesh. But when the two come together, we get some lovely change in structure that allows for it to go through and become a nuclear transport receptor. In other words, import factors plus cargo leads to a change in structure. That change in structure leads to an ability to now interact with the mesh, making it more permeable to cargo complexes, therefore allowing the necessary cargo to enter the nucleus. We can really boil this down to just being a specific example of how a change in structure leads to a change in function. So, to sum up what we've learned so far, nuclear pore complexes are kind of like gates. I say kinda because NPCs are always open, but in order to get through the gates, the cargo needs to flash some kind of ID or proof of VIM, very important molecule. That proof comes in the form of a karyopherin, which is a nuclear import factor, and the combination of the cargo and the import factor makes a complex that's able to disrupt the mesh network that usually prevents molecules from entering the nucleus. Bada bing, bada boom, and now we have necessary proteins machinery that are inside the nucleus. So, now that we know how cargo is actually able to get into the nucleus, we have to slow down and ask what's directing it into the pore. Nuclear pore complexes themselves don't determine the directionality of import, so what does? How do cells know which direction molecules need to go? Well, we've learned that it often has to do with concentration gradients of molecules. Recall the case of the sodium-potassium pump, in which sodium and potassium are moving from high concentration to low concentration. The gradient that we're going to be focusing on is the RAN-GTP gradient. A little bit about the protein itself. So, RAN is a protein that binds with GTP, and GTP is similar to ATP, just with a guanosine instead of an adenosine. There's a couple of key players involved in the overall process, but we should focus on RAN-GDP and RAN-GTP. The concentration of RAN-GDP is greater in the nucleus than in the cytosol, and RAN-GDP is more highly concentrated in the cytosol. So, the concentration gradient is actually maintained by two more RAN proteins, a guanine nucleotide exchange factor and a GTPase activating protein. Note that the exchange factor is in the nucleus and that the activating protein is in the cytosol. Okay, now that we've named all the players, it's time to see how they actually interact with one another. So, RAN, when it's bound to GDP, is capable of binding to the karyopherins, therefore giving us a complex of RAN-GDP plus a karyopherin plus cargo, and that complex is able to go through the nuclear pore. So, once we're in the nucleus itself, RAN-GEF, the exchange factor, comes in and adds a phosphate to the GDP. This addition causes a change in structure in the karyopherin such that the cargo dissociates from it. So we can kind of see why it's being called an exchange factor, because we're trading the cargo for phosphate being bound in this case. So, our cargo is staying in the nucleus while RAN-GTP and the karyopherin move out into the cytoplasm, where the activating protein, RAN-GAP, activates the protein complex and causing hydrolysis of the GTP back into its GDP form. And now we're ready for another round of import.
Alright, I bet you didn't think there would be so much information about small pores on a tiny organelle in a minute cell. It certainly must make you appreciate the various intricacies of life. So to summarize everything we have gone over, the nucleus is a control center of the cell where lots of vital processes take place. The protein machineries required for those processes are assembled at the ribosomes in the cytoplasm, but get into the nucleus via nuclear pore complexes or NPCs. Nuclear pore complexes are protein structures embedded in the nuclear membrane, which act as the gates into the nucleus. These gates are always open, but selectively let in only proteins with an NLS tag bound to nuclear import factors called karyopherins. NPCs don't themselves determine directionality of import complexes, but it's a RAN-GTP gradient that does. We hope you enjoyed this video and learned a thing or many.